He recently joined the Department of Molecular Microbiology and Biotechnology here at Tel Aviv University in the Life Sciences faculty. And he's gonna describe his new research on engineering living cells for expression of artificial materials.
Hi everybody, thank you for the invitation and the opportunity to show my research. So I'm going to talk about how we can use living cells for the production of materials, how we can engineer cells, manipulate cells for the production of molecules that don't need to be something natural. And so it's taking you a different away from what you saw this morning, but we still need the same techniques, the same methods, CRISPRs, genomic engineering, and all of these kind of things in order to manipulate the cells. Now, why we are doing such things? So there is mainly two reasons. The first one is we obviously provide new opportunity, new in vivo applications. The second one is to provide accessibility of the molecules. If such molecules is expensive, having the biology doing the work for us, this fantastic bio machinery is then will reduce the price and have everything accessible. Let's start with different kind of example of what the nature knows to do. So what you have here is a bacteria that know to create and synthesize magnetic particles. So the black parts here are magnetic particles. Why they are doing such things? Through the evolution, it enables them to survive and to go deep into our cells. Here you have the diatoms. So what the diatom is doing is creating on the cell wall the silica nanostructures. And you can think about the world of biology, trees, other materials, and especially the spider silk, for example. And think about how we'll be able to create the same spider silk, but without the spider. Maybe a yeast can do the work, maybe bacteria can do the same things. And then the accessibility of this kind of fantastic material will be produced. But what we need? So we need genetic writing and genetic editing. So we need techniques like CRISPRs, cloning, Gibsons, and so on. We need to manipulate many genes together. We need to control where each they are going to be activated and expressed and at which time. So what you need to have is such genetic circuits that it is synthetic biology. So synthetic biology is a field that starts since 2000s with the idea to provide a new language to manipulate cells. So what they create or they synthesis different kind of libraries of genetic parts, promoters, RPS, ribosome binding site terminators, and so on. Such you have a lot of libraries, you just have the possibility to bind them in a different way, so you can have your program, you can synthesize the DNA, so obviously if we are here in the conference from synthesis, you can directly buy keyblocks and have all of your DNA already synthesized. And from here, you just transform all of this that meat, all this genetic circuit that you need to have into any kind of cells. So it can be bacteria, yeast, human cells, if you want to do some therapeutic compound. So this is the point of synthetic biology. As I said, as well as CRISPR, so you may want just to introduce plasmids that have already the circuit, or if you want to go to the genomic part, you will use CRISPR to edit and change the genomic part of each one of these cells. But let's look at an example of how does it work. So this is a very simple genetic part. This is a promoter. When it is activated, the RNA polymerase recognize it and then transcript and express the protein. We can add another part upstream that is called, that will remove, not allow the RNA polymerase to bind and then prevent the transcription and the expression of the protein. And you may have any kind of signal that we want today to control this very simple genetic part. So you may, in this case, you have the signal and it will activate again the RNA polymerase. So you have today, as I say, any kind of possibility to do it. Light, pH, temperature, ions, molecules, anything that you want to activate this kind of genetic circuits. For example, here, if you increase the light intensity, you will express more and more of this protein that is activated through this promoter. And here is, for example, the bacteria plate. When you light precisely where you want, it will express the pigment, and you can see the picture in this case. So during all of these years, what synthetic biologists have done, they create all of these kind of tools. They provide library. Why we need library? Because we don't want to have recombinations in the cell, so we need to have each one of these parts with a different sequence. So we have a lot of promoters, we have a lot of RBS, we have a lot of terminators, and even if you go further in the very recent work, you can just go in the software, design to the circuit that you want to have, and it will automatically provide you the precise DNA that you need to order. So if even someone that is not expert in synthetic biology can today go in the software and order exactly the circuit that he wants. But I'm here, so what I'm interested in to do is nanomaterials. So I'm coming with a material that is from the chemistry world, from the physical world, and material science. I will look into the biology, any kind of biological function that will help me to create these materials in the cell. I will then take it into the genetic circuits that will activate, control the expression of each one of these parts, use the resource of the cells, this is RNA polymerase, ribosome, everything that the cell is giving to us, and create these materials. And this is a material that I'm interested in. So it's going to be a little bit confusing. I'm interested in creating DNA. So not DNA that is creating our nature, but I'm interested in DNA that is doing these kind of things. So we have a field that is called DNA nanotechnology. It starts since the 1980s. And it gives us the possibility to use oligos. So short oligos, about 14 nucleotides, hundreds of oligos, and you can detect exactly the structure that you want to get. Very precisely, and this is making this material very important material in nanotechnology, since you can control at each place of these nanostructures to put anything that you want at the nanoscale. And for it's just an example of how does it work. So this is our DNA, they are oligos. You will program them to assemble exactly in this way. You will have this very specific junction. You can have sticky ends and they will create two-dimensional patterns. You can go and create more complicated crossover junctions, and you have the weight on the top and the weight on the bottom, and the blue one is bringing these two DNA together. So it's not just utilizations that you know, just duplexings, but it's combining duplexings in a very complex architectures. And you can see here, any kind of structures can be created. All of the structures are about 100 nanometers, and you can take smiley face, letters, whatever you want, very precisely. You have even software that you can just design the structure that you want. Tell you to buy the oligos. Again, you can go to IDT to buy these oligos, and you put them in the tube and get the nanostructure that you're looking for. Different kind of applications come out. The first one, as chemists, they use it as a catalyst. So you can put everything where you want. So you can put enzymes or different kind of organic molecules and lead to very specific reactions. You can control and use it for material sciences to control nanoparticles, to place, dynamically control the nanoparticles, so even grow the same way that we are cooking at home. So you are baking at home, so you can just grow into these nanostructures, into the DNA nanostructures, silver and gold particles. Or, more attractive for the biologists, this can be used also as a drug delivery systems, when you can put the drugs in the middle of these nanostructures. And only when it will be close to the cell, it will open himself and release the drugs. But what is missing, and this is going to my research, is how we are going to do this kind of things in the cell. How we are going to do it genetically. So we want everything to introduce new parts in the cell, plasmids, and what we want to get out is all of these nanostructures. Now the challenge is obviously what we need first is how you pass from this genetic circuit that we'll construct and engineer, induce the cell and create single-strand DNA. Single-strand DNA is not the natural material. So we have nucleotides in the cell, but we don't have DNA in the form of single-strand DNA. We have duplexes, plasmids, chromosomes that duplicate. But how do we pass from here to it? So this is the first challenge. Then the second one is, can we assemble DNA and create nanostructures, go to different kind of applications, or if it's used to reduce the price, we purify the DNA from the cell and create the nanostructures. So since the challenge, this was my research that I made at MIT during my postdoc. So again, let's start with the first challenge. How do we precisely express single-strand DNA that have precise 5 to 3 points? They should have any nucleotide that we want. We can change any sequence, so there is don't need to tune again and again the genetic circuit, the program to create a very specific DNA. We want them to have any sequence that we want, and we want also to have multiple DNA together. So circuit that will have 4, 5, 6, 100 of oligos at the same time. So, as I said, what you need to do if you're going and looking to create a certain material in the cell, you will start to look what biologists are doing and what else the biologists enable to do, which kind of function they can use. So the one that I came out was the HIV. So whether the virus is working is introducing an RNA in the cell We have also an enzyme called the reverse transcriptase. It's taking this RNA, reverse transcripting back to DNA, integrate it into the genome, and you have the infection. So there is a way the human cell to move back from RNA to DNA. And if you look into the molecular level of how this mechanism is working, so you have the viral RNA that is here, you have a tRNA that is coming from the cell, is bind to the blue part, bind together, so enzymes or reverse transcriptase recognize this part, bind to it, will start to polymerize DNA on the RNA, so the very special function to make polymerase not on the DNA template, but on an RNA template. Then you have the second function, the same enzyme, you have also the function of RNase H. You will remove the RNA from the DNA RNA template, you will do it 2 times, you will create 2 double-strand DNA, integrate into the genome. Now this function, I'm working in bacteria, and my goal is to do it in bacteria to the fact that I want to have this material accessible. Now how do we bring these things into the bacteria? So if you look in the literature, the reverse transcriptase from the HIV has not been found to be working in bacteria. So we need to find the reason why it's not working, and we need to engineer it into the bacteria. So the first place to go is maybe what we don't have in the bacteria is a binding part that leads the reverse transcriptase to bind to the substrate. So what we have again, we have a tRNA and the VRNA that is on the bottom. So what we don't have maybe is this blue part. So what I did is that I conjugate the 5' and the 3' to be together. So this is the same picture that you just saw before. The 5' and the 3' will be conjugated together. In this way, we will have an RNA that will be terminated with this tRNA. So the tRNA maybe is missing in the bacteria, so we need to have it in the bacteria. And what we want is also to specifically bring the enzyme only to its substrate, not to any kind of RNA. We want to reverse transcript only the RNA that we want. So in this case, in order to know what is the terminator, if we have the RNA polymerase, when we start the transcription precisely on the terminator part. So this guy here will be a terminator. So the first thing was to engineer a terminator. So how do we look at if this sequence can be a terminator? We have cassette that we have a GFP and RFP. We put in between these genetic parts of this sequence, and if it's terminated, we have low fluorescence of RFP and high level of GFP. So here, if you want, you have the results. If you don't have anything here, any terminator, you will have also expression of both GFP and RFP. This is the expression when you have a very strong terminator, natural terminator for bacteria. And this is my guy here, the tRNA binding part for the reverse transcriptase, and you can see that it can create and is enabled to be a terminator. So the first part of the engineering will have now the missing part of the tRNA in the cell, and it will be engineered as a terminator due to the fact that it will give us selectivity towards the RNA that we want to work on. The second thing is, the question is, does the enzyme really work with it? Does the enzyme really bind to this secondary structure? So we put on the second cassette that when we have an RFP transcript and express, so the transcription of the mRNA will be terminated with HTBS. So the HTBS is this specific tRNA binding part. When we let the enzyme to the reverse transcriptase, it will start to polymerize on the mRNA, and then it will reduce the fluorescence, it will reduce the expression of the GFP. And this is exactly what we saw. So this is a regular terminator, and if we have our terminator at the end of the mRNA of the RT, in the presence of the RT, you can see reductions of the fluorescence. So we know at this part 2 things. We can create a terminator binding part, that is a binding part for the HIV, and we know that HIV is binding to this place, and even doing something, and reducing the fluorescence. So how does the production pathway will look like? So we'll have a genetic part, synthetic genetic part, that will create synthetic radicals, will transcript RNA, the RT will bind to it, we'll start to polymerize DNA on it, we'll do it DNA-RNA template, we'll reverse them, we'll switch himself to the RNase H, and in this case we'll remove the RNA from the bottom. What we'll have at the end of the cell, a reverse transcription of this guy. So the RNA that is here free will become DNA. Still at the end, we still have an RNA binding part that is still staying there. When you will purify, you always have RNase A that will want to purify plasmid or anything that you want. It's very cheap, and you will have precisely the 5.3' sequence to DNA. So all of this process is right here. And I really like this slide because this brings the reality of the research. It didn't work. So when I purify the oligos, I knew that I have this binding part, I knew that it's doing something with the fluorescence, but when I try to purify the oligo from the cell, I got nothing on the chair. So everything looks perfect. So as a mechanism, this is what I wanted to have, but nothing came out. So here I was stuck. 7, 8 months. It's a lot of time just to close the project and just go away. But no, I really wanted to do it, so I stay here. So what is the reason? What do we find at the end? So there is a problem in this situation that we don't find any DNA produced and expressed from the cells. So there is two places. The first one is maybe really we have the terminator, this we know. We know that it's binding to it due to the fluorescence activity that decreases. So maybe just creating a small piece of DNA, and then we have an RFP that is not fully fold, and a small part of the RFP is missing, and this is reduction of fluorescence that we see. The second possibility is that the RNase H is not working on the RT. So we have this part, but what we don't have is the removing of the RNA from the DNA template. So what the idea was, let's look at the way that the RT-PCR is working. So I'm sure that you're all familiar with RT-PCR. The way that is working, we have an RNA, you bring an oligo. This oligo have any sequence that you want. It's not specific to any RT. So in this case, we have already an oligo that is created on it. So if I would add another reverse transcriptase in the same time with other one, we will continue the process here that it can bind to the DNA. And if the problem was the RNase H, so this is a medium RT, they will remove the RNA from the DNA templates. So in this case, I will have a solution for both of them. And what comes out is that we have DNA only if we were put in the cell and co-express the HIV RT and the merinocalia in the cell. And the only piece that you can see here, this is the DNA. If you put only the merinocalia, so you will be from here, you don't have any DNA due to the fact that you don't initiate any kind of reverse transcriptase in the cell. So from my point of view, this was a breakthrough of my work, because you enable us to understand why the HIV didn't work in the cell, what was missing in the cell in bacteria, and now I can produce single-strand DNA. So from here, I just will check and create more structures. So this was, at this moment, I was really happy. So in this case, since everything is genetically encoded, so you can control it with a sensor that I just described before, with lights or small molecules. In this case, it's IPTG. And if you have the IPTG, you will express of the single-strand DNA. If you don't have IPTG, so you will not have single-strand DNA. The next stage is to control and to check the quality of the oligo. So to check with other commercial oligos. So this is commercial oligo 72 nucleotide from IDT. And if you purify my DNA from the bacteria, you can see exactly the precise bands that appear. If you do change into genetic parts, the size of these parts is 49, the band will go down and be exactly the place that you need to be. But this is not enough to have quality of this molecule. What you need also is to check the sequence. So in this case, we use sequencing and also deep sequencing, next generation sequencing. We count these bands, we use different kind of methods to enable only the sequencing of single-strand DNA, so this is a method that is already published. And we were able to really see exactly the precise nucleotide that is exactly on the genetic part. So we can create oligo, it's comparable to always commercial oligos, and we can sequence it. For me, it's just to create nanostructure. So if you want to create nanostructure to reduce the price, you will create oligo, you will purify it from the cell and create a nanostructure. So in this case, I engineered one DNA that may assemble to himself and create a very long DNA nanostructure. So you can see it exactly here. This is a chain. You have a lot of this structure, all of these pens here are the nanostructures. And it's very similar to what you have when you use natural synthetic chemical oligos. The second one was just to use the same oligo to add 10 nucleotides to it, and you will create two-dimensional nanostructures. So you can see besides the 10 nanometer squares here that are here, so you can reproduce exactly the same things that people are doing using chemical oligos with these oligos that come from the cell. So the next challenge is to understand if we can create these oligos and assembly in the cell. Because oligo may assemble in the condition of intracellular conditions. This is a question that we don't know since we didn't have the possibility to put single-strand DNA expressed in the cell. So in this case, we have these genetic parts, circuits, that will express 4 single-strand DNA together. They are engineered to assemble one to each other to create this crossover junction. These are very fundamental structures and architecture things that show it in the beginning that we can create, if we have these structures, any kind of structures that we want. So first I purified it from the cell. At the end, we still have the RNA parts that enable me to visualize this through the FM. So you have these X nanostructures. But this is not enough. I mean, if I purify it from the cell, it's quite possible when I'm purifying the DNA nanostructures, it's assembling together. So yes, I can express 4 single-strand DNA in the cell. Maybe when I'm purifying the DNA from the cell, they assemble one to each other. So the question was to create a sensor that we visualize and detect if my nanostructure is disassembled in the cell. So in this case, we co-express split white field that will assemble, will bind to our nanostructures. Only 4 of them, 4 of the DNA will be one which shows up. So in this case, you can see if you have expressed only 1 oligo, you don't have fluorescence, 2 oligos, you don't have fluorescence, 3 oligos. Now 4 oligos generate full hybridization of the 4 parts together, you have full fluorescence. And in this part, I'll just control if you just make mutation on the zinc fingers, that is the zinc fingers that bind the fluorescence protein to the nanostructures. In this case, you will not have any kind of fluorescence. Or if you don't have the HRV, so the reverse transcriptase that is doing all of this, you will not have a fluorescence. So there is really 2 things that I just described. First of all, you can count with from another field that is not biology, a molecule that has nothing to do with the biology. You will look into the biology and use the bacteria to create it. And the only reason that is possible is the fact that we have all these synthetic biologists, genetic, and that enable everything to be done very fast and very cheap. So now we can look at what we can go from here. So now that we can do structures in the cell, we can use it as scaffolding. We can do exactly the same thing that chemist was doing to control precisely place of molecules and activate and catalyze better reactions. We can do it by DNA in the cell. So advantage to have it done by DNA as compared to RNA, for example, in the fact that DNA, single-strand DNA, has nothing to do with the natural process of the bacteria. So you can knock down into the genome all the exonuclease, all the SOS parts, and have a bacteria that is, that will enable the single-strand DNA to be non-integrated into the cell. So it should provide us a better opportunity to do scaffolding as compared to peptide or RNA that is self-degraded to the natural process. Second thing is DNA has been used also to deliver drugs. So if we have it expressed in the cell, we'll be able to sense if the cell is sick and then to express it into the cell. So we have a sensor. You just don't come with the drugs and the drug will kill any cells. If you have it controlled into the genetic circuits, we'll be able to control the therapeutic compound according to the signal in the cell that are sick. We can even further use it as an imaging. DNA has been used to image and process into the cell, and the advantage of it again into the cell is obvious. Then the second part of where we can go is to create materials that are not completely obvious. What you can see here is the phage. So what they've been doing with phage lab at MIT, they use the phage as a template to grow inorganic materials, to bring energy. So with this phage, they were able to activate a car. So the battery of a car can be made of phage. So what they are doing it is we can have a lot of phage and produce a huge yield of phage. They engineer just the protein and the peptide that are on the cell of the phage to bind and to grow inorganic material each time to each other. In this way, if I would do it by DNA, I would be able to create any structures that I want. So I would be able to do better structures, better phage, made of DNA that was not possible due to the fact that you don't have the possibility for nanostructures in the cell. With this, I will summarize my talk. So what I described today is the ways that we can engineer single-strand DNA in the cell. We found the way what was missing in the bacteria to enable it and expression in the cell. So the first one was the missing tRNA. The second one was the need to have co-expression of the 2 RTs together. And we can even use this method to make regulation to the fact that if you put this binding part for the reverse transcriptase at the end of an mRNA, it will regulate the mRNA expression of proteins. With that, I need to thank the people that made it possible. So first, Professor Chris Voigt, who was my PI at MIT, my collaborator Peggy from Harvard, my founder, that was Andrew and IT, that was a postdoc award from IDT, that's here. And I'm looking my lab here, very close to here, to building from here, the green building. So if someone is looking and motivated to do this kind of research, they're welcome to contact me.
Questions?
Thank you very much for your excellent presentation. I want to ask you if you make these materials made with the oligo of the DNA, which are the characteristics in compared to the materials you can find nowadays in the industry?
The oligo is exactly the same as the oligo that you will get from synthesizer, the chemical synthesizer?
Yeah, so, or the materials that you can find nowadays in cars or construction, like, which are the advantages, like, in the delivery perspective?
I mean, so if your question, I mean, first of all, we can reproduce the same molecules, right? So quality is exactly the same, the sequencing is the same. The problem that we have is the yield, you asked me, of the productions. So my goal is to arrive to the yield of the phage, so whether we have phage, genomics, and all these parts, so this yield should be the text. So for now, we're not there, but this is things that we can do through simple evolutions of the proteins and all of these kinds of things. But at least for now, we have the process that you know where to go.
No, but the question is, it was maybe more durable or maybe more flexible than other kind of materials. Like if you can...
It's exactly the same materials. I mean, this is the same molecules, exactly the same things. There is no difference.
Thanks.
Do you see any their response to this foreign DNA, because it's different from the foreign DNA that bacteria see, which could be plasmid, which is circular, or even single with the specific ends? So do you see any response to this new DNA that the cell is seeing for the first time?
Your question is the cell are toxic.
Any response?
When the response? For now, toxicity, they are not toxic. So, I mean, the way that you're, so what is toxic is more the expression of the urinochemia. It's a little bit toxic, so you need to control it to put it in a low protein plasmids to reduce the toxicity of the cell. But what we have here, I use a cell that is a knockdown the possibility of the SOS and of the exonuclease. So it's many competent cells like TH10B that I'm using. So a lot of this kind of response are already inhibited from the cell. The other cells that are natural cells, I don't